Thank you, Jim, for the warm introduction, and thanks for inviting me to come down. It's a pleasure to finally come to Virginia Tech, and thank you, everybody, for taking time out of your day to come and listen to my talk. And so this is the title of my talk, and it's really going to be the second half of the talk, which is our recent work in collaboration with Jim's group looking at microRNAs in parasitic plants. But before I get there, I want to spend some time introducing the general system and then talking about some of the other things that we've been doing in the lab for the last 12 years. And so when we think about RNA interference and small RNAs, we're really talking about this paradigm where short guide RNAs become bound to these proteins called Argonaute proteins. These are proteins that are found in almost all eukaryotic organisms. There are some oddballs that have lost the system, but in general, most eukaryotic organisms are doing this. And these short RNAs don't encode any protein. They serve as specificity determinants for these Argonaute proteins to interact with target RNAs and essentially cause repression of some kind or another. And exactly what happens depends upon the context and the organism and the specific Argonaute, but it's a repressive activity. And where we see these coming from are really two sources. And so there are single-stranded precursors that have some region of self-complementarity, and so they fold back upon themselves to make a stem loop. And that stem loop is digested by these endonucleases in the Dicer family or Dicer-like family to produce an initial duplex, and one of the two strands becomes bound to the Argonaute. Or they stem from a long, truly double-stranded RNA where the two strands aren't covalently linked. And very frequently, particularly in plant systems where I do most all of my work, that double-stranded RNA is synthesized by an RNA-dependent RNA polymerase that acts upon an initial single-stranded RNA. But they all kind of funnel into this process. And these are relevant in a number of contexts, and probably, although it's not a focus of my research, and this will be the only virus slide that I have in there, probably the most relevant way in which RNAi works, particularly in plants, is as an antiviral system. Okay, so this is a classic image from David Balcom's 1999 paper. This is the first detection of any small RNA in any system. And what it is a northern blot of infected Nicotiana tabacum leaves with potato virus X. And it's being probed with a probe that has homology against the potato virus X genome. And what you see is over the time course of infection, as the virus replicates, you get more and more of these short RNAs that have homology to the viral genome. And if you look at systemic leaves, that is leaves that were distal to the initial site of virus inoculation, you see the small RNAs accumulating there. And this is important for antiviral resistance. In fact, every plant virus that's been systematically studied that actually is an infectious plant virus encodes a protein whose function is to suppress the RNA interference response of the plant in some way. So in other words, in order to be a successful virus, it seems like you need to be able to shut down or evade or redirect the RNA interference system in plants. It's also curious, so Nicotiana benthamiana, which is shown here, this is one model system that plant molecular biologists use a lot. It's been used particularly by virologists for the last 80 or 90 years. It's an Australian species. It's related to tobacco. It has nicotine in it. Apparently, Aboriginal populations do chew on this. You do get some nicotine out of it. But it's been a good model system for virology because a great number of viruses infect this organism. And it turns out the reasons that viruses seem to infect this thing is because it's a natural mutant in one of these RNA-dependent RNA polymerases. And so these images on the left are wild-type Nicotiana benthamiana infected with 4 different viruses. And the images on the right in each picture, which show healthier plants, are benthamiana where that defective gene has been complemented, I believe, with the Medicago version of that RNA-dependent RNA polymerase. And so that's just to emphasize the real importance of the system in terms of responding to viruses, right, and silencing the viruses. Another venue in which the RNA interference system is particularly important is biotechnology, and in particular, the introduction of foreign genes into stable transgenic plants. One of the persistent problems in that process is transgene silencing. That is the observation that very frequently when you put a gene into the plant, even though the DNA is there, it won't work. Okay? You don't get expression, you don't get the protein production. So this is a classic image from 1990 from Rich Jorgensen's work. These are petunias, these are purple. This is a publication from 1990, so it's kind of pre-PDF. I apologize for the the thing. But so petunias have purple flowers, right? And so the experiment here was to introduce an extra copy of chalcone synthase. It's the rate-limiting enzyme in anthocyanin production, with the expectation of increasing the pigmentation of this flower and perhaps, depending upon the promoter, turning the leaves purple as well, so increasing anthocyanin. But in fact, what's frequently occurs is you get these sectors in the flower that lack all anthocyanin. And sometimes they're symmetrical, sometimes they're asymmetrical, sometimes they're complete absence of all anthocyanin. And so what happened here was you put an extra gene in, and not only did your gene not work, didn't make more purple pigment, it inhibited the function of the endogenous gene, right? So this phenomenon was what's sort of called post-transcriptional gene silencing, because it was shown that it occurred post-transcriptional. People refer to this as co-suppression. And long story short, this is really all modulated by RNA interference. The genome has essentially detected that this transgene has come in, mounted this RNA interference response. Small RNAs are produced that both target the endogenous gene and the incoming transgene. So this is still a bugaboo, this sort of transgene silencing. And it turns out if you go and do deep sequencing and looking for small RNAs across plant genomes, they're quite numerous. Endogenous small RNAs are numerous. That is, without a viral infection, without the introduction of foreign DNA as a transgene, they're just all over. And so this is a screenshot from Arabidopsis. And so for size perspective, this is a protein coding region here. This is like a 42 kb arbitrarily selected region of the genome. And these data here are small RNAs that are expressed. And so you can see there's a bunch of them. They're mostly intergenic, and that turns out to be true. So one of the important classes of endogenous small RNAs are microRNAs. And we define microRNAs essentially by the fact that they're coming from these precursor stem loop structures. And they're diced by this dicer protein very precisely to make a single duplex. Typically, that duplex is two 21 nucleotide microRNAs, and one of the two becomes bound to an Argonaute protein. And these interact with messenger RNAs. And so typically, they're interacting with a messenger RNA, causing it to be chopped in half, also causing the translation to be repressed. It's a combination of the two methods. They can interact with long non-coding RNAs and cause degradation. And there's some sequestration, interesting things that can happen. But essentially, this is the major activity. These microRNAs come. They're part of endogenous gene regulatory circuits that are required for the proper control of certain messenger RNAs by turning off their transcription, or turning off their productive protein producing capacity. So these are quite relevant, as evidenced by the drastic phenotypes that you can observe. These are either Arabidopsis or maize phenotypes that are due to perturbations of microRNAs. I won't go into the details here, but suffice it to say that one of the reasons is that many of the endogenous microRNAs, one of their frequent targets are transcription factor, or mRNAs that encode transcription factors, and those transcription factors are sort of master regulators of plant development. So you can get these very, very drastic pleiotropic phenotypes due to changes in organ identity, changes in juvenile to adult transitions, and things like that. This one's particularly interesting. So this is maize, mutant maize. This is normal maize, corn in other words. Even non-plant biologists here probably know what corn normally looks like, right? Upright, big thing. So this mutation here, yes? I could take you on a field trip later. One of the things I did when I used to pick sweet corn. So yeah, it's horrible. So anyhow, this is a drastic mutation. And so this is an allele called corngrass that was characterized in the 1950s and was sort of a touchstone of maize genetics at the time, because it was one of these example mutations where the phenotype of the organism had changed so drastically that people could imagine, hey, look, this is how new species form, right? And so that's one of the classic debates in evolution. How do you get genetic changes that change the architecture of the organism so drastically to have a new form? This mutation turns out to be a transposon that jumped into the promoter of a single microRNA and caused that microRNA to be overexpressed. That microRNA regulates a transcription factor that controls developmental timing. Basically, when do you stop making leaves and start making flowers? And this mutant just basically never stops making leaves. It's a little bushy maize plant. It will make a tassel, and those are the flowers, by the way, reproductively eventually. So development's a big deal. And microRNAs are well studied and they're quite famous, but they're actually not that numerous. Okay, so if you look across, these are 3 well-studied plant genomes, Arabidopsis, maize, and rice. If you look at microRNA genes in the genome, it's on the order of a couple of hundred that have been annotated from each species. If you compare that to protein coding genes, right, we're talking 100-fold less, right? So protein coding genes are on the order of 20,000 to 40,000 for these organisms. But it's even worse than that because there's a lot of misannotated microRNAs. And so if you boil it down to the ones that we think are really high confidence, it's very few. Many of those loci are paralogs of each other. That is, there's multiple copies that encode the same exact mature microRNAs. So if you boil it down to families, it's very few. And if you boil it down to the ones that are conserved, it's not that many. Okay? So microRNAs are important, but the conserved ones and the high confidence ones really aren't that numerous. Nonetheless, we spent a long time working on microRNAs, and one of the first things that we've done is sort of taking advantage of this. So one question that comes up is, get bored standing on one side. So one question that comes up is, how do you go about finding all of the targets of a given microRNA? And so we realized early on that because the Argonaute protein cuts messenger RNAs in half, that we can detect these downstream remnants after the cleavage. And the reason for that is, so for messenger RNAs, the poly-A tail is the major determinant of stability. So if we cut this thing in half, it no longer has a cap. It's not going to be translated anymore. However, these remain somewhat stable in vivo, so we can go find them. And so you can do this one at a time. This was pioneered by Jim Carrington's group 16 years ago now. So these little denominators here are just cloning 5' ends of uncapped mRNAs and determining, hey, most of them end here. That's exactly the 10th nucleotide of complementarity to the microRNA. And we know biochemically that's where the Argonaute proteins will cut. So we adapted this to high-throughput sequencing. So we basically make RNA-seq libraries where the input is polyadenylated 5' monophosphorylated RNA. So that includes all of these sliced products. And if you sequence deep enough, you see things like this, where this is positioned along the mRNA, this is the frequency, and you see one giant peak here. And if you map where that is, it ends up being precisely in the middle of a predicted microRNA target interaction. In other words, it's pretty convincing evidence that that's a real target. So we call that degradome sequencing. A lot of folks use this now. And the challenge turns out the libraries are simple to make. It's the informatics, because it really is degradome sequencing. We're sequencing basically all of the little degraded fragments of mRNAs that are in the cell, and there's a lot of them. So pulling out signal from noise and finding a reliable way to get p-values is the key thing. And so we have software online that folks can use to do this. We've worked with it. So one of the other areas where we've made, I think, some significant contribution is understanding the rules of how much base pairing between a microRNA and a target messenger RNA is enough to trigger silencing, right? So perfectly matched things will definitely work. Absolutely random matches, right? 1 out of 4 nucleotides, they definitely won't work. But where in the middle, do you get a threshold effect, right? So how do we know what's a target? So I use this color coding in the next slide. So it's a colorblind, acceptable color coding. So basically, blues are Watson-Crick pairs. The little orange color here is a GU wobble, and the magenta is a mismatch. So we developed a system based on these sort of transient assays where we transform the leaves of Nicotiana benthamiana with agrobacterium and sort of in a medium throughput way go through these. So we made a dual luciferase sensor, and we mutated target sites in one of the luciferase genes, and we can measure mRNA accumulation and protein accumulation. And the protein we use luciferase activity as the proxy for protein accumulation there, and measure the activities. And so this is just one figure from that paper. And here we're marching through from either end of the microRNA target site, making longer and longer consecutive mismatches. And if you see data that's way out here, these are target sites that work, so they repress the target. If you see data that's on this dotted line, the long dotted line, this is the 0 line. So these are things that just don't work at all. And what you can see here is that there's a big difference between the 3' and the 5'. That is, you can get up to 4 consecutive mismatches at the 3' end, and it works pretty good. If you get even 1 mismatch at the 5', it kills it most of the way. In 2 mismatches, you're done. So there's a difference in the site specificity of this. And through a bunch of assays that I won't take the time to show you, we really have this sort of rubric now for understanding plant and animal microRNA targeting data, and that is there's a difference between plant and animals in terms of how their microRNAs target messenger RNAs. And so for plants, we really need this region 2 through 13 to be paired. It will tolerate a single mismatch in there with some drop in efficacy, but if you get more than one mismatch in there, it doesn't, in our hands, it never works, so we don't see natural examples of that either. The region in the 3' of the microRNA doesn't really matter so much. It tolerates several mismatches. The first position never matters, and I'm pretty sure based on crystal structures of animal argonauts, that's because the Watson-Crick face of the first nucleotide is just completely buried in the argonaut protein, so it's not available for pairing. And that's different than in our bodies and in the bodies of other vertebrate animals. MicroRNAs really only require positions 2 through 7 to be targeted in order, or to be paired in order to function. So there's a fundamental difference between the two kingdoms there. OK, and so I started this sort of part of the talk with this screenshot, the genome browser, showing that these small RNAs are really widespread in the intergenic regions of the genome. But then I told you, oh, there's not that many microRNAs, right? So what the heck are the rest of them? And so one thing that's been motivating a lot of our work recently is this issue. And so this is a review article that my student and I published. And I think it's a really interesting graph. So what we did here is just take publicly available mRNA-seq data from protein coding genes from Arabidopsis flowers, align it against the genome, and then just ask, well, how many nucleotides of the genome had a certain amount of RNA-seq coverage? That is, how much of the genome is involved in making messenger RNAs? Okay? That's about 34 million nucleotides, about 25% of the Arabidopsis genome. Okay? So we did the same thing with small RNA sequencing data from flowers, and it turns out it's about 12 million nucleotides of the genome. It's about 10% of the Arabidopsis genome, and there's not much overlap between those two positions. Okay? So there's quite a bit of genomic real estate that's dedicated to this system, and it's not the same genomic real estate that's dedicated to protein coding gene production. Okay? And then if we ask, well, who's annotated? So for mRNA-seq data, this circle is this blue circle over here. Here's where we have RNA-seq data. This one is previously annotated genes, and this is Arabidopsis thaliana, so it's a very well annotated genome. And you see there's very, very good overlap here. This large crescent, this is just the introns, right? So they're part of annotated genes, but you don't have RNA-seq data there because introns are spliced out, messenger RNAs. This little thin thing, these are unannotated, novel transcripts, maybe the 5' UTR, the 3' UTR is a little longer, something like that. But there's pretty good concordance. If we do the same thing for microRNAs, there's not that many microRNAs, there's this whole universe of things. And so that's motivated our studies to really understand this. Not so much to understand it, but to map it and to have stable reference annotations. Where are these genes? So we know what these things are. So there's this whole other system of endogenous small RNAs in plants where you have an RNA-dependent RNA polymerase. It makes double-stranded RNA out of these short little precursors. They're chopped into predominantly 20-formers instead of the 21-mers that you see for microRNAs, bound to particular argonaut proteins. And what these things seem to do is hone in on these nascent transcripts. So this is in the nucleus, and this is an alternative DNA-dependent RNA polymerase. And these bind here, and they recruit chromatin remodelers to the vicinity of where they're targeting. And these little red dots are meant to represent DNA methylation and also repressive histone modifications. And so this is a system essentially where small RNAs will direct DNA methylation and repressive chromatin modifications onto the plant. And you particularly see this for transposable elements and things like that. We've done a little bit of work on this. And so if you're paying attention in my earlier slides, the other argonauts that I've shown you, I said that they interact with messenger RNAs and they catalyze the cleavage. They cut that RNA in half. But in this model, it's hard to understand why the argonaut would need to cut this thing, because the model is basically a proximity model, right? So it's dragging it to the region of the chromosome. But we've proven that it does slice things. And so I won't go walk you through the nitty-gritty of this, but this is basically small RNA sequencing data. And all of these clusters of small RNAs disappear in an Argonaut 4 mutant. These are just biological replicates. If we make transgenic plants where we complement that AGO4 mutant with a wild-type Argonaut 4, most of them come back. You can see the red colors. But if we complement it with a slicing defective version, they don't come back. Okay, so there is slicing going on in the system, and this is recent work from Steve Jacobson's lab using nascent transcription, and they could find this signature that's consistent with slicing. We don't understand why, but this is the way it is. Okay, and so I want to emphasize this slide a little bit because this is sort of the summary of our knowledge of the molecular biology of the small RNA pathways. We have microRNAs. They interact with targets. We have these so-called heterochromatic siRNAs, which are very, very numerous in the genome. They direct DNA methylation at the bottom here. And then in the middle, we have these secondary siRNAs. And so what happens is sometimes these interact with a target, and this remnant that we clone with degradome sequencing becomes a substrate of an RNA-dependent RNA polymerase. Double-stranded RNA is produced, which is diced up into small RNAs. And those small RNAs can feed back into this pathway. So you have a positive feedback loop here. And they can also feed down into this heterochromatic siRNA pathway. So even though I'm guilty of it, and many people in the field are guilty of it, of sort of describing, oh, microRNAs are different than these things, they really are connected. We have examples where one flows into the other. And this is, as I said, my work is focused on endogenous small RNAs. But a lot of this probably reflects the antiviral capacity of this. That is, you have a post-transcriptional branch that feeds into a co-transcriptional branch, particularly probably for retroviruses or pararetroviruses that integrate into the genome. So what do these 24 nucleotide siRNAs do? Without belaboring the point too much, they're basically repressing transposons endogenously. So if you, this is not work from my group. This is from Keith Slotkin's group, and this is from Becky Mosier's group. Essentially, these things maintain DNA methylation at transposable elements. And so these mutants here, RDR2, POL4, these knock out all of those 24 nucleotide siRNAs. And you have these big defects in transposon methylation. In Arabidopsis, you don't see much of a phenotype if you get rid of this system. But it's most likely because the Arabidopsis genome is very, very free of active transposons. There's not much active transposition in there, so it doesn't matter much. This is recent work from Becky's group at Arizona in Brassica reba, right? When they knock out the 24 nucleotide siRNAs in Brassica, they see a big defect in seed size set, and it's probably due to rampant transposition. So a lot of our work has been focused on methodology, and in particular, we've really wanted to make sense of all of this deep sequencing of small RNAs and have a piece of software, which we call ShortStack, that will take the small RNA libraries, take a reference genome, do proper alignments of it. And we published a paper on the alignment, just the alignment problem alone, because one of the issues is you have these little short RNAs. Yeah, aligning it's trivial because you just look for a 21 nucleotide match. But the issue is you often have cases where it could match multiple locations in the genome at a very high frequency. And you have to make decisions. So how do you treat those, right? So we came up with an algorithm that makes decisions that are better than random guessing based on the biology of the small RNAs. I don't have time to go through it, but you can read the paper, or if you're interested in small RNA analysis, you can use the code. So it's up on the internet, and you can use it. And the whole idea here is that we make sensible whole genome annotations of all of the endogenous small RNAs, and we make our best effort at classifying, oh, this looks like it's a heterochromatic siRNA, this looks like it's a microRNA, this looks like it's junk, because we get a lot of junk as well, so degraded bits of ribosomal RNA and things like that. So one thing we've done recently, this is work from one of my students, Seth Polydor, is look at mutants in Arabidopsis that essentially eliminate this pathway and this pathway. So according to the canonical sort of understanding, everything that we're left with should be microRNAs. And the end result is that there are actually some things that are unexplained. So it's a small number, but we do have some small RNA loci that don't really fit into this paradigm. They tend to come from genes. They tend to be less conserved. And it's pretty much all we know about them. So we tried hard to bang on it. But it does come out of this understanding of both building the tools for doing the sequence analysis and having these mutants available in Arabidopsis. And the other big project that we're still right in the middle of is sort of using our small RNA annotation tools, the software that we've developed, and then leveraging the huge investment in genome sequencing and really the ton of free data that's out there. So there's a ton of small RNA sequencing data from multiple plant species. And systematically in multiple plants, annotating all of the small RNA genes in a consistent manner, using the same software, same settings, and things like that. So basically, we have 41 of these species annotated. We have about 12 billion small RNA reads. So we're very proud that we have more small RNA reads than human beings on the planet. Pretty cool. Pretty cool. For now, well, you know, are we really going to break 12.4? That's a big number. We'll have to do more sequencing. And this should be coming out soon. So the challenge is here, besides the data curation aspect of it, is we're trying to integrate these data so that it's not just, oh, you look at soybean and there's a soybean genome browser and that's it. We're trying to connect it across species so we can readily make user-friendly interfaces that allow you to ask, oh, how conserved is this? And that's involved. It's been challenging, but we'll release this probably this summer. OK, and so I want to talk about the project with Daughter and some of the progress we've made there, the more organismal and newer side of my research, which is obviously a collaboration with Jim here, and also with Claude de Pamphilis, who's my colleague at Penn State. And these two fellows are the ones who really got my interest up in this and sort of started this. And so I think many of you may know this from your exposure to Jim's group, but just in case you don't. So Daughter is the common name. Cuscuta is the genus. There's, depending upon the source, about 150 or 200 different species of daughters around the world. And these are plant parasites. So these are parasitic plants that have make their living by attaching to other plants. These are stem parasites. They're obligate parasites. They need a host in order to complete their life cycle. They're hemiparasitic. They still do a little bit of photosynthesis, right? So there are these papers where people observe oxygen being evolved. There's chlorophyll in the organism. But they're quite reduced, right? So they're just these little vines. This is a tomato seedling, by the way. This is an Eppendorf tube, and this is a seedling of Cuscuta growing out of it. It wraps around the stem, makes these haustoria that project. So this is a cross-section where the parasite is here, the host stem is here. This organ is called a haustorium, and it goes in and it siphons off nutrients and water and carbon and everything else. The organism doesn't make leaves. It doesn't make roots anymore. Okay? So all of its water is coming from the host. Nearly all of its carbon is going to come from the host because it's not making leaves. And it ramifies into these sort of golden vines that end up sort of wrapping around the host and eventually flowering. Most or all of the species are self-fertilizing weeds, so they make a ton of seed when they get on a host. And they're really, they're really interesting organisms. They have a lot of funny names. So if you get on Wikipedia and just look at common names of it, yeah, there's some good ones in there. Hailweed. So one of the neat things about it is, particularly the species that Jim's lab has been working with, and then by extension my lab has been working with, is that they have a very broad host range. So they'll attach to a broad number of species, including under lab conditions Arabidopsis, which means we can do genetic manipulations and transgenics and look at mutants and things like that. OK, and so the organ that it uses is this organ that is unique to parasitic plants called a haustorium. So this is in the case of Cuscuta, it's a projection that comes off of the Cuscuta stem. It burrows through the host tissue, certainly degrading cell walls, and gets in and hunts out vascular bundles and connects either directly or indirectly to the vascular bundles in order to get the carbon. And so this is an outreach video from Jim that I shamelessly stole because it's gorgeous. And I know because this is home turf, maybe you guys have seen this before. But if you haven't seen it before, it's kind of remarkable to watch how this parasite grows and moves through its life cycle. So it germinates like any other plant. So it's a eudicot, right? Throws up the two cotyledons and piles up. This is time-lapse, by the way, in case you guys aren't. Yeah, it doesn't move that fast. You don't have to run. I've had people ask me that before. It's fine. So there's data out there that shows that they can smell their hosts. So they smell volatile organic compounds that come off of plants, and they can direct their growth toward host stems based on that. And you can see once they contact a vertical surface, they twine. Okay? And this is a response essentially to touch. And they have to do this rapidly because the seeds are obligate parasites. Like I said, they don't even make roots. So all of that water that they're using in that initial phase seems to be just from the drinking up of water from the seedling stage. When you get a successful connection, it burrows in. You can see the swelling of the haustoria over the course of 12, 24 hours, something like that. And this, once it's done, this will be the primary connection. Okay? And so it actually senesces everything off below that point of connection. It can regrow multiple vines from the site of attachment. Each one of those new vines is going to start doing that dance again and twirling and smelling new hosts and new locations in order to attach to.
Are those pluripotent cells? I mean, if we cut them, they grow again?
Yeah, they are pluripotent. You can propagate it by cuttings, and we routinely do that, and so does Jim's group. Yeah, and it's not complicated. It's a pair of scissors, and you cut a 5-centimeter chunk, including the apex, off, and you just throw it on another plant, and it'll coil up and go again. And if you let it go, it gets pretty wild. And if you observe it in nature, actually, we've done some collections locally at Penn State, and I'll talk about that data at the end of the talk. But you just drive around and you see those clumps of sort of golden clusters and weedy disturbed areas, and it's Cuscuta. It's very common, actually. So the species we're working with it's native in the lower 48. You can find it everywhere. Yeah. And so I got interested in this largely because of these observations. This is particular to Cuscuta, but for many plant parasites, including fungi and also parasitic plants, it's been shown that if you engineer a host plant with an RNAi inducing hairpin, that RNAi activity, that gene silencing activity, can move into the parasite. Okay, and this has been proposed as a possible way to control the activity of the parasite. So you, in this case from Sinha's group at UC Davis, they targeted a gene in Cuscuta that is required for the shoot apical meristem to function. So the meristem is the part of the plant that develops new organs. So if you kill the meristem, it's going to stop growing, right? And indeed, that's not quite what they saw. They slowed down the growth with doing this. But the fact that this works suggests that either these hairpin precursors or the small RNAs themselves, or both, are able to move across the haustorium, right? And to me, Jim, and Claude, this suggested, well, if you can engineer the system, probably nature is doing this already. The channels are open in order to move these things around. So we went looking for it. So the hypothesis was that there are these naturally occurring small RNAs that are moving one way or the other during daughter infections. And so essentially, Jim and Gunjun in his lab gathered RNA samples from Arabidopsis stems, and we sequenced them, we meaning Sima in my lab, sequenced them and analyzed them. So our experimental setup was pretty simple. So this represents Arabidopsis stems, and what Gunjun did was wrap Cuscuta around it, and we have 2 replicates each from these 3 different regions. So this thing we call I, this is just a mixture of Arabidopsis host stem and the haustorial region of the parasite. So the RNA is really a mixture between the 2 species coming out. We call this parasite stem. It's the apical region of the vine. It's parasite only. We call this host stem. It's a few centimeters above the part of attachment, and it doesn't include, at least not intentionally, it doesn't include any Cuscuta tissue. And so what we did was look for small RNAs that were induced in the interface relative to either the host stem or the parasite stem, because presumably those might be the most important ones. And so it really worked out if we compared interface to parasite stem here. So if people are used to looking at RNA-seq data, this is an MA plot. If you aren't used to it, basically this axis is the overall abundance of the small RNA locus. And this is a ratio. So if the dot is up here, it's higher in the interface. If a dot is down here, it's lower in the interface relative to parasite stem. And you can see most of the data are lower. And that's a dilution effect, right? Because our interface sample is a mixture of RNAs from both species. And the plot that I'm showing you is only the Cuscuta ones, right? So by definition, pretty much everything looks like it's lower because it's being diluted with host RNA. But nonetheless, we see a bunch of things that are upregulated. And a number of the things that were upregulated were clearly microRNAs. And so these are just 2 examples of predicted microRNA secondary RNA structure hairpins, and the color coding is based on depth of small RNA sequencing coverage. And so you can see these sort of red regions here. This is where most of the read comes from. It's a very typical microRNA pattern where there's precise processing of one region of that hairpin. And we can confirm that by northern blots and reproducing it, that they're quite specific to the interface. CS here means control stem. So this is Arabidopsis stem that's just not been parasitized at all. So it's not some feature of Arabidopsis. And the thing that we noticed about this informatically, you can see that I've separated them by size. So the blues are 21 and the greens are 22 nucleotides. And that was surprising because most plant microRNAs are 21s. And the ones that are 22s, although rare, they had been observed. But when you do see a 22-mer, it was known that it really favored this secondary siRNA pathway for whatever reason. So in other words, most 21 nucleotide microRNAs, they cut their target, and this gold thing never happens. But if you have a 22 nucleotide microRNA, it was known that, yeah, it's very likely that this fragment will funnel into this pathway, make these secondary siRNAs that then feed back and do more and more of this. And so when we saw those data, we thought, well, if these microRNAs are being produced at the haustorium, perhaps they're being produced to target host transcripts. And if so, perhaps we would expect the targeted host transcripts to show this secondary siRNA signal, which we would expect to result from these 22 nucleotide microRNAs. That turns out to be true. So this is one example. This is the SIOR1 gene in Arabidopsis. This is host stem, interface, and parasite stem. And the y-axis is just showing small RNA sequencing coverage. This little line here represents a putative microRNA target site where it's a microRNA from Cuscuta. This is the messenger RNA from Arabidopsis. And we can see, yes, indeed, specifically in the interface, we see these abundant small RNAs. The downstream is very typical. These have properties of secondary siRNAs, which I won't go into, but basically they're mostly 21s and 22 nucleotides long. They're arranged in this so-called phasing pattern because they have this defined end, and so this really looks real. And we can find several examples of this. And so the BIC1 gene, Arabidopsis BIC1 gene, has 2 different microRNA target sites, 2 different Cuscuta microRNAs, clear evidence of secondary siRNAs. Same with this gene down here. And then these 3 genes are all related to each other. They're very closely related to each other. And for plant folks, these genes encode the receptors that perceive auxin, one of the major plant hormones. OK, so we find several Arabidopsis messenger RNAs where there's clear evidence of targeting. So we can go and directly look. So I told you earlier about these assays where we do degradome sequencing, or we look for these cleavage remnants. And that's what these little fractions are here. So we went and sequenced uncapped messenger RNAs from these interfaces. And indeed, we find clear evidence that they're being sliced. This is just the gel showing that. So I is interface, CS is control stem. We see bands in interface, but not control stem. And when we cut them out and clone them, they all work. This one where you see both is a positive control. This is not related to the Cuscuta system. It's just an endogenous microRNA. If we look at messenger RNA levels, as we would predict, if these are really being targeted by Cuscuta microRNAs, the messenger RNA levels of these targets are decreased in the interface. So control stem and interface, 8 or 9 biological replicates. The only one that didn't work was this one. So the data were pretty noisy here, so there's no significant difference. But for BIC1, SIOR1, TIR, and AFB2, and AFB3, it looks good. This is a control gene, by the way, just a housekeeping gene to show that it's not some general artifact of our sampling that everything seems to go down. So that's as you would expect. So one question is, so it's known from Jim and Claude's previous work that Cuscuta actually, it takes in a lot of large macromolecules from the host, including messenger RNAs. Okay, so you can find intact host messenger RNAs that move into Cuscuta. So a question might be, well, are the things that we had observed, these microRNAs that target host genes, is that just reflecting the fact that these messenger RNAs got sucked into Cuscuta and then it's maybe perhaps not relevant? And so an indirect way that we thought of to ask this question was taking advantage of the fact that we know in Arabidopsis the genes that are responsible for making secondary siRNAs. So in particular, Dicer-like 4, and this little thing you can't read here, the purple thing, this is SGS2. Okay? So if we grow parasites on these mutants and ask, well, do we find these secondary siRNAs? We don't. Okay, so this is wild-type Arabidopsis. This is one of these secondary siRNAs, and we see it in wild-type. It completely disappears in the SGS2 mutant. It goes down a lot in the Dicer4 mutant, and these are all controls. And so the fact that those secondary siRNAs seem to depend upon the host's Dicer and the host RDR strongly implies that at least that amplification is going on inside of a host cell, right? You couldn't imagine, well, you could, but it would be like, I don't know, imagining the sun revolves around the Earth, right? Even when you look at Mars's motion. So it's a harder explanation, right? So it's indirect, and I should say that one of the things we're really working hard on is making a more direct assay of this to prove that the microRNAs go in by directly visualizing the microRNAs by in situ hybridization. It's tedious. We haven't traditionally done those in my lab, so we're working toward that. But I think there's good evidence here that it's happening inside the host. So the key thing is, okay, does it have a biological significance? Okay, can you demonstrate that it does? And we've done that. So one way we could do this very simply, because it's a Arabidopsis, because these target genes that we've identified are all known genes that have known phenotypes, we could just get the mutants, either from the Stock Center or from the labs around the world who have worked with those genes. And if our hypothesis is correct, if you grow Cuscuta on these mutants, our hypothesis is that these genes are being targeted because down-regulating those genes somehow confers an advantage to the parasite, right? So we would predict that growing Cuscuta on those mutants would increase the fitness of the parasite in some way. So we did that by growing Cuscuta on these various mutants. Col0 here is wild type. So this is the typical sort of amount we get. And we can see in 2 of the cases, SUR1 right here and AFB3 here, we have a significant increase in Cuscuta biomass when growing these mutants. Another 2 cases, no, we don't see any difference. And this one goes the opposite way of our prediction. And the explanation for that is pretty simple. So the BIC1 mutant is a very sick plant. So it has a pleiotropic development. So basically it doesn't make good stems. The stems fall over. The parasite has a really difficult time attaching to this thing. So I'm not sure how meaningful these data are. So, right, 2 out of 5 doesn't sound great, except when you sort of stop to consider, the argument I would make if you're talking about parasite or pathogen host interactions, the idea that you have a single virulence target that has a directly measurable effect in the context of knowing that the parasite makes many, many virulence factors, right? So we have a couple that by themselves make a direct effect. Actually, I think that's pretty remarkable. OK, so Arabidopsis is not the host that Cuscuta evolved on in nature. I would bet you money on that. So the things that we found in Arabidopsis, if they're meaningful, one would expect that the true hosts, which is a very broad host range, would also retain those target sites. And so one way that we can indirectly ask that question is go into sequenced plant genomes, find the homologs of all of these different target genes, and ask, do these homologs retain complementary sites to these Cuscuta microRNAs? Is it plausible? And I told you before from our previous work, we have a really good idea about the amount of complementarity that you need to make a functional microRNA target site, right? So this is the result of that. So we had 31 really well annotated udicot genomes at the time we did this analysis. So for BIC1, for these guys, about half the time we find a good target site, a little bit less for this gene. For the TIR and AFB ones, most of the time we do. And GAPDH is a negative control here, just to show informatically how frequently we would get false positives. And it looks pretty convincing. So yeah, many other genomes have plausible complementary sites. And then we could directly ask the question by repeating the experiment, except growing Cuscuta on a different host. This time we grew it on Nicotiana benthamiana, because that's what we had in the lab. And it attaches readily to Nicotiana. Repeat the small RNA sequencing. We find very similar signature of upregulated things. And if we ask, are these upregulated things the same as what we saw in the original experiment? Indeed, they most of the time are, including the handful of things that we had directly shown target Arabidopsis genes. Okay, so it looks like the induction of these microRNAs so far, based on this one experiment, it's not specific to a given host. It seems like a generic response where it cranks these things on. Okay. And I don't think I have the data. Oh, I do have the data later. Never, sorry. Non sequitur. Please.
So this parasite plant non-specifically target other plants? I mean, does it matter which one is going to be the host plant that's going to provide all these treats?
It has a broad host range. I could say that. I don't know, and Jim certainly knows a lot more about this than I do, but I can't immediately think of really well-controlled experiments where people tried to do host choice kinds of things, because it seems like when the parasite germinates, it's got attached to something immediately. Okay, so whatever's nearby. Yeah, I'm not sure I answered your question though. Sorry.
No, it's okay. I was thinking because of the 30 microRNAs that you found in common. Right?
Yeah.
I mean, like they need to be represented in all
Yeah. I mean, I can hand wave on it. And so the hand waving answer would be, I have a sense that these are just all microRNAs that might be useful on some host. But on this particular host, we only find evidence of these 6 or 7 targets. Maybe on another host, we'd find different ones. The other thing that I would say with regard to that is, so far, I hate when people do this, but I'm going to do this anyway. I'm going to flip all the way back here. We've been looking for things like this. That is, where the incoming microRNA was a 20-tumor, and it triggered the secondary siRNAs, which gives us a really easy indicator to see when there's a targeting event that's happened. A lot of the induced microRNAs we find are not 20 tumors, they're 21 mers. So I think there may be more targeting going on than the stuff that we've really harvested the low-hanging fruit here. So I think between those two things, I think we have more targets than we think in the organisms where we've looked at so far. Plus, yes, I have a hunch that it just produces a bunch of these, not all of which will work on any particular host without any discriminatory, yeah, if that makes sense. OK. So this is a neat experiment, too, that I wanted to share with you. So one question is, are these things, so this is just showing Nicotiana benthamiana and Arabidopsis. Yes, same things. We could confirm the sequencing by northern blot. We also did this experiment where we germinate seedlings and allow them to, cruelly allow them to wrap around a piece of wood. Okay, they're going to die. But they will differentiate these pre-haustoria. You can see these little bumps here. And if we isolate RNA out of those pre-haustorial bumps don't express these microRNAs. And so there's some indication that so that's this lane right here. pH means pre-haustoria, right? So there's some indication that this requires contact with a living host, perhaps a metabolic cue, perhaps just water, I don't know. And we're sort of following up on that. This is just showing in Nicotiana benthamiana, yeah, we find homologs of the same genes making these secondary siRNAs. So all that makes sense. So what are these targets? So I hinted at it before, but I'll give a little more detail here. So tier 1, AFB2, AFB3, these are related and partially redundant genes. They encode F-box proteins that are required for auxin perception. So they couple perception of the plant hormone auxin to transcriptional responses downstream. How does that connect to plant parasitism? I do not know. Auxin does lots of things in plants. There is a literature for plant bacterial interactions that modulate auxin signaling. But I can't give you, it's not a just so story with this. It's a conserved element of this parasite, but I don't know why. The same with this one. So this target is a gene called schizoryza, and alternatively, a different group named it heat shock factor binding 4. So this is a transcription factor that is thought to be a transcriptional repressor, and it came out of mutant screens in root development. So this is wild-type roots of Arabidopsis color-coded by stem cell identity. So the stem cells of roots are here in the quiescent center, and their daughter cells differentiate either to become vascular tissue or cortical tissue or endodermis and things like that. The phenotype of this mutant is there's an extra layer of cortical cells in the root produced. So how does that connect to the stem and what the parasite is trying to do? I wish I could tell you, but I don't. This one makes more sense, intimate sense. So this is a gene called BIC1, and on this diagram, it's this little orange thing right here. BIC1 is a kinase whose mutant phenotype, besides being stunted pleiotropic plants, it's exquisitely sensitive to a number of different pathogens. So it came out originally of a screen for plants that were sensitive to the fungal pathogen botrytis. So BIC1 means botrytis-induced kinase 1. But it's hyper susceptible to bacterial pathogens and to fungal pathogens, and it's a signal transduction molecule in pathogen defense. And so that makes some sense, perhaps, why the parasite might want to dampen down pathogen perception. This gene SUR1 is a phloem protein, and so the major cell type in the plant phloem are called sieve tube elements. And again, for non-plant folks, phloem is the vascular tissue in plants that conducts sugars, essentially. Okay? And when the parasite attaches, it's certainly getting sugars out of this. And so this phloem protein accumulates in these big agglomerations. So this is a GFP fusion looking at a row of sieve tube elements in the root. And this is out of paper from Mike Knobloch's group. So these P proteins are extremely abundant, and they're thought to be involved in sort of wound responses and controlling the amount of photosynthate flow in the phloem. So this makes sense to us, too, why the parasite might want to target that. So we started to do a little bit of work. So Mike Knobloch's group at Washington State had a CR1 YFP fusion transgenic Arabidopsis plant. So you can see the sort of green here is our transgenic YFP expressing in the sieve tube elements. And if we attach Cuscuta onto it, we can see Cuscuta lights up with YFP. And we expect that because we know that Cuscuta takes up large macromolecules. A day later, it's completely cleared from the host. Okay, so these are the same images, just under different illumination. You can see the host stem is here. It's completely gone. And so we have some indication that it's taken up by Campestris and also cleared from the host, consistent with this microRNA targeting. So the last little bit I want to tell you about is an evolutionary story and really kind of a funny tale. So we started doing these experiments, and it's actually difficult to tell what species you have. And so we had a lot of confusion about different seeds and who's growing what in which lab. We eventually figured out we've been working with Cuscuta campestris from Jim, and it came out of a tomato field in California. And we had another isolate that was collected locally by Claude that turns out to be another species, Cuscuta granovii. Okay, so this phylogeny is a small snapshot of the larger species phylogeny based on plastid and ribosomal DNA markers. And so granovii and campestris are somewhat closely related, but they're clearly in different clades, okay? And they're distinguishable. So we did the same experiment with granovii. We did small RNA sequencing, asking which small RNAs go up. And similarly to campestris, we find 100 or so small RNAs, mostly 21 or 22 nucleotide long, that are induced in the interface relative to the parasite stem. But surprisingly, if we ask, is there any overlap, there's very, very little. Okay, so these are the things we found in campestris. These are the things we found in granovii. It's only 2 of those couple hundred things that are in common between the 2. So in other words, the microRNAs that campestris induces are largely different in sequence than the ones that granovii induces. I'll spare you all the details, but we've done the same things with granovii. We have identified, so these things we collect in the field, they'll grow in Arabidopsis in the lab, which is great. It's another broad host range parasite. And we've identified targets in Arabidopsis that are clearly being cleaved, and they're not in common. Okay? The only one we find in common so far is CUR1, which is targeted by both of them. These are the ones that I got done telling you about. These are actually some new ones that didn't make it into our published paper, but with further study, we see these are targeted by campestris. And these are granovii things. So one of the interesting things we observed, so there's 2 things interesting here, right? So these closely related species seem to have almost completely different microRNA repertoires. And the targets we've been easily able to identify are largely different, okay? Which suggests, I guess, rapid evolution. But if you know these genes, so BIC1 is a signal transduction gene during pathogen defense. MAP kinase 3 is a signal transduction gene within pathogen defense. And actually, they're in the same pathway. So this is not my work. This is from people who spent a long time looking at these signal transduction pathways. So BIC1 is this receptor-like cytoplasmic kinase. And one of the things that it does is funnel down through this MAP kinase cascade to target these nuclear genes in pathogenesis. And indeed, MAPK3 is targeted by Cuscuta granovii microRNA. So it seems like there might be evidence that even though these microRNAs perhaps have changed rapidly throughout the course of the diversification of Cuscuta, that there's these recurrent themes, particularly this defense pathway, perhaps the phloem, the CUR1 thing is in common. And the last data slide I have here is probably the most intriguing. So we have right now 2 different populations of Cuscuta granovii. So this was collected by Claude 11 years ago, and this is the one where we did the small RNA sequencing. This is one that I collected last autumn from a different location near Penn State. It's clearly Cuscuta granovii. And we've done just qRT-PCR looking at these small RNAs that we found from this sequencing experiment to ask whether the second population has the same ones. And the answer is yes, yes, no, no. Okay, so we have some evidence that there's even population level differences in the types of microRNAs that we're going for, which I think is going to be, once we get this sort of sorted out with a bit of genome sequencing and things like that, it would be an interesting story. So that's all I've got. So the end part of the Cuscuta story, I think we've shown that these microRNAs go in and target host messenger RNAs. The secondary siRNAs probably serve to amplify it. At least 2 of the handful of targets that we've tested so far in Arabidopsis have a clear effect in restricting Cuscuta growth, because when we make the mutants, Cuscuta grows more. The induction is not host specific. Okay, so this is basically the same microRNAs are expressed on at least 2 different hosts. We think it's probably plausible across the host range. Granovii also does this, but uses different microRNAs. And our overall hypothesis is that these microRNAs may be functioning somewhat similar to classic virulence factors, that is, to modulate host gene repression in order to benefit the parasite in some way. All right. So with that, I want to thank the folks who did this. So this is our funding sources over the years. Unfortunately, not all at once, but over the last 13 years. And really, the people who I'd like to highlight are collaborators on this Cuscuta project, particularly Jim, and also Claude at Penn State, and all of the folks who work with them. And then Saima, who really drove the Cuscuta project in my lab. She's since earned her degree and moved on to a postdoc at Keith Slotkin's lab. And then Nate, who's a current graduate student who's done a lot of the more recent work on Cuscuta granovii and also so that. So thank you for your attention. I'd be happy to take questions if there's time.
We've got a couple minutes for questions.
So I kind of lost you there, and all of a sudden you had some candidate target genes. Were there lots of candidate target genes?
Yeah.
Was it a hit parade and you chose the top, or what?
It's definitely a hit parade, and I chose the top. So I could say what we've done is there's a bunch of candidate targets that you can find bioinformatically. And then we took that list and looked in the sequence data to attempt to find ones where there was the secondary siRNAs. And so far, those are the only ones we followed up with. Yes. So, and in fact, the question I had in the middle there was somewhat similar. I think there are more. And we may have to look harder to find them because they may not be making this obvious signature of secondary siRNAs. And there are certainly many more microRNAs that are upregulated. So there's, yeah.
So then the other question would be, you have something to do with phloem function,
Yeah.
something to do with transcription.
Yeah.
Is it possible to do a time course?
Yes.
What happens when?
We're doing it. Yes. Yes. The answer is it's absolutely possible. We just haven't done it. Yeah. Well, it's not. It's more just setting up the experiment and priorities. Doing the sequencing time course would be expensive, but now that we know the sort of proxy microRNAs to use, we could just do QRT or something. Yeah.
I have two questions. So you presented the microarray as like a degradation factor, but oftentimes in mammals, at least, these microRNAs bind and they don't degrade; they just sit there.
Yeah.
Sort of interrupt the ribosome.
Yep.
Does that happen in plants at all, or have you seen that, or considered measuring the protein output to see if it changes?
Right. Pretty much no, actually. So there's a big difference. And so in animals and in humans, most of the microRNA target sites are in the 3' UTR. And the 3' UTR is a unique region in the mRNA because it's the only region that's not funneled through the ribosome, right? So, and it's been shown in those systems that you need stable, persistent interaction of the Argonaut microRNA complex to have any kind of repression. In plants, no. The target sites are everywhere, and it doesn't seem like there's any need for stable. In fact, if you engineer, I didn't talk about it, but there's sort of a third class of targeting sites, if you looked carefully in some of those slides, where there's a big central mismatch in the middle. If you put that into a plant, what happens is the Argonaut microRNA complex will bind to the target, but it's unable to cut it, and it's unable to release. And when that happens, the cell recognizes that and just degrades everything. So the microRNA gets degraded and the target gets degraded, it all goes away. It ends up acting as a competitive inhibitor. So yeah, so I think there's pretty good evidence that you don't require or generally use this sort of persistent, stable interaction in plants the way that you do in animals.
Okay. And then I have one more question. So if you were to do RNA-seq after you either express microRNAs or knocked out their expression, because those fragments are still left behind,
Yeah.
you wouldn't see much of a change, right?
Yeah. Well, with RNA-seq, you would because you leave the 5' fragments actually degrade very rapidly because they don't have a poly tail. But actually, in the olden times, which all these students you're too young to remember this, used to be this thing called microarrays, right? And you couldn't do it because that happened. These 3' remnants would be still present in the sample, and they would hybridize to the spots on the array, and they would give you the indication that the mRNA level didn't change. But and those, the classic microarrays were 3' biased. The probe sets tended to come from the 3'. So, yeah, you have to be careful. And actually, when we do those experiments, if we're just measuring targets by QRT-PCR, we're very careful that the primer sites we have one is on one side of the microRNA cut site, one's on the other, so that we're only measuring uncleaved messenger RNA. Yeah.
Do you ever compare the difference between those two?
We have in a few cases, and there's a big difference. Yeah, yeah, yep. Go ahead.
I'm curious about the evolution and the divergence of the small RNAs produced. You saw the two different parasites had very different secreted populations.
Yep.
Which is presumably due to Red Queen. You know, the plants are evolving to counteract that. Do you see the same thing in your survey of all of the plant small RNAs between different?
Yeah, that's a good question. The answer is yes, actually. So if you look at sort of microRNA populations as a whole, the sort of typical thing you find from a regular plant is 12 or 20 really abundant microRNAs that are really well known and very well conserved across all land plants. And then you find another couple hundred that are variable in abundance, sometimes more tissue specific, lower abundance, that don't have any obvious conservation with the data that we have. And so there's been interesting back and forth in the literature about what that means. And actually in the past, I've written things that said most of these are non-functional, right? Because there's no obvious selection, there's obvious purifying selection, there's no obvious conservation, and it turns out it's very hard to find targets for these things usually. However, now we've seen cases where, in this work with Cuscuta, but also in a few other contexts, of these apparently less conserved microRNAs clearly being functional. So yeah, I think this is common. I don't know if this trans-species thing is common, but the idea that you can have rapid diversification is common. It's also, I would say, unless you have absolute conservation of the microRNA, it's actually very difficult to determine conservation because you're talking about 21 nucleotides, right? So and the hairpins, yeah, they all come from hairpins. The hairpins aren't constrained except for the requirement to fold into a secondary structure. So, and that's pretty loose. So you can have, G's and C's flip and so reconstructing the history of these genes is, I think, pretty challenging. Yeah, so knowing if they're old or not is hard. Yeah.
Yeah,
a couple minutes. One more question, and then we'll break. Yeah.
Just kind of mapping these 21 nucleotide pieces back to the genome is basically getting lost in the sea of noise here. So have you also tried kind of mapping the, I mean, it's basically like, we have the same issue with making primers specific.
Yep.
Do you try mapping these also to like a transcriptome and then kind of comparing the two and seeing, if you can find more, targeted gene targets there?
Yeah, it doesn't work, and it's because both the microRNAs and the endogenous siRNAs, they actually get diced co-transcriptionally, so extremely rapidly, right in the nucleus. And so the precursors pretty much don't accumulate. So you don't find them in, if you do transcriptome reconstruction, you really don't find them. Yeah. Otherwise, yeah, that would be the solution. But yeah.
All right, good.
Thanks.
Well, we're out of time, so thank you.